everyone, welcome to OsteoBytes. My name is Christina Iptoma, and I am mom to Osteo Angel Dylan and Director of Scientific Programs at MIB Agents. And today on OsteoBytes, we are talking with Dr. Dan Regan, and he will discuss his OutSmarting Osteosarcoma funded work on Focal Adhesion Kinase Inhibition to improve Losartan-Sunitinib Immunotherapy in Metastatic Osteosarcoma. Thank you so much, Dr. Regan, for joining us on OsteoBytes today.
Thanks for having me.
Yeah, we're thrilled to have you, and it always brightens my day when I get to spend some time of it with Vicky, who is our panelist today. Vicky is an osteo warrior and a member of our MIB Agents Junior Advisory Board. So a little bit more about our guests, and I'm just going to say that there are a lot of long syllable words in your bio, Dr. Regan, so I'm going to try to make it as best I can.
Sorry.
Dr. Dan Regan is an assistant professor at the Flint Animal Cancer Center at Colorado State University. And as I mentioned, he's one of our OutSmarting Osteosarcoma 2022 grant recipients. Dr. Regan received his DMB degree from the University of Georgia and subsequently completed his residency training in veterinary anatomic pathology, the PhD in the Department of Microbiology, Immunology, and Pathology, which we'll refer to from here on out as MIP, at Colorado State University. And in 2018, he joined the Flint Animal Cancer Center and MIP in the College of Veterinary Medicine and Biomedical Sciences at Colorado State University. The focus of his lab is to increase our understanding of the interplay between the immune system and the non-immune tumor stroma, and how these compartments of the tumor microenvironment promote metastasis, as well as respond to and mediate extrinsic mechanisms of resistance to anti-cancer therapy. And to investigate this area of cancer biology, his lab utilizes the combination of in vitro three-dimensional tumor cold culture models and animal models, focusing on breast and osteosarcoma. Dr. Regan also has a strong interest in comparative oncology and leveraging naturally occurring cancers in dogs as both a surrogate and intermediary model to evaluate and validate his lab's investigations into the tumor microenvironment. And in collaboration with the lab of Dr. Steve Dow, his lab conducted immunotherapy clinical studies in dogs with spontaneous osteosarcoma, and these study results have led to a phase 1 clinical trial in children with osteosarcoma. His lab's continued long-term research goal is to fully elucidate the mechanisms by which tumors prime non-malignant host stromal cells of distant organs to promote their metastasis and chemo-resistance in order to identify novel targets for host-directed stromal therapies, which poison the soil for effective combination with conventional tumor cell-targeted drugs. Whew.
Wow, right. That was, I don't know if I need to talk after that.
Okay, I'm ready to hand it over to you. Okay, welcome, Dr. Regan, and welcome to everyone joining us today. Please feel free to add your questions for Dr. Regan to the Q&A. And this is definitely meant to kind of be a conversation. So feel free, you're always welcome to hold questions at the end, but feel free to also ask questions throughout. And we can ask Dr. Regan during his presentation. So why don't you first hand it over to Vicky to introduce herself?
Yeah, so hi everyone, my name is Vicky Hoy, and as Christina said before, I'm a member on the Junior Advisory Board and I'm an osteo warrior. I'm currently a freshman at Villanova University studying finance, and thank you for joining us today, Dr. Regan.
Okay.
And I'm so sorry, Dr. Regan, I totally forgot. I was so excited to introduce Vicky. I forgot we had some exciting announcements to make. So let me make those and then I will finally hand it over to you. So we have our Turbo Virtual Tumor Board next week on the 9th. So if you're a clinician or researcher with an interest in osteosarcoma and you want to be added to the calendar invite, or if you're ever interested in submitting a case, just fill out the form on our website. I'll put a link in the chat. If anyone's in the Houston area, we are going to be there on November 30th in the evening for a mingle, and I'll put more information about that in the chat. And some super big news, which is so great, and it just happened to be serendipity that we have Dr. Regan with us today, but we just opened our 2023 OutSmarting Osteosarcoma RFA today. So you can find more information on our website. Applications are online on Proposal Central. The due date is February 3rd this year, and we'll put more information about that in the chat as well. So with that, Dr. Regan, we'll turn it over to you and feel free to start sharing your slides.
Thank you, Christina and Vicky, for the introduction and for having me today to discuss our work, and definitely excited to talk about this. So All right, we will jump. Okay, so kind of before we get started on some of the lab's work, I just wanted to provide a little background. Many of you already may be familiar with this concept, but really, kind of as Christina talked about in my introduction, the overall focus of the laboratory is on the tumor microenvironment. So this collection of non-malignant, resident, and recruited host cells, which can sort of extrinsically influence and promote cancer progression and metastasis. And so this concept was really introduced in the late 1800s by a British surgeon, Stephen Paget, who retrospectively evaluated the medical records of approximately 800 patients with breast cancer. And he made a pretty astute observation that, when these patients died, they were dying from metastasis, and it was to very specific organs. So he would see metastasis in the brains or the bones or the lungs, but wouldn't see it in other sites like the kidney or the spleen or skeletal muscle. And so he postulated basically that there must be some specific characteristic or composition of these secondary organs that receive these metastatic cancer cells, or the seed, i.e., that promotes this process. And so he called this the seed and soil hypothesis, and this microenvironment being the soil. And so, I think for many, many years, essentially a century, this hypothesis was relatively discounted, and people thought that the process of metastasis was purely by chance or just more anatomical in nature of tumor cells lodging in smaller vascular beds. But really in the early 2000s, I'd say, the work of David Leiden's group, Rosandra Kaplan, a number of people really started to spearhead work on this tumor microenvironment and elucidate some of these mechanisms. And now we know that certain soluble derived factors from cancers that are in primary sites like the bone or the breast will circulate, and they'll prime these distant organs and start to condition the soil. And so this is really what we're interested in here. And what I'm showing here in this video, which doesn't look like it wants to play, but also in this EM image here, are tumor-derived exosomes. So these are small particles, nano-sized particles about the size of a virus, that carry a lot of biological cargo. So DNA, RNA, proteins, and these can effectively function like hormones, circulate, leaving the primary tumor, being secreted by those primary tumor cells in the bone, if you will, traveling through the blood, and then acting on target cells in these distant sites to help prime metastasis. Oh, there we go. So yeah, here's some of these, a live image of these particles that we isolated in the lab. And so we're really kind of focused on three primary cell types of this microenvironment, and again, of the lung specifically. So I think, my interest really arose in this during my research and pathology training because of that work coming out of David Leiden's lab, and specifically a paper by Dr. Kaplan in 2005. And I was really focused on osteosarcoma from the beginning. So I think, the fact that this tumor has this really strong tropism, if you will, to spread to the lungs and metastasize to the lungs, provides a unique opportunity, but also a potential therapeutic vulnerability to kind of study that lung soil and understand how it's mediating osteosarcoma metastasis. And so in our approach, we're heavily focused on three cell types of that soil. So the first one we're going to talk about today is this population of CCR2 expressing monocytes, or what we call inflammatory monocytes. But we'll also focus on lung fibroblasts, resident lung fibroblasts there, and as well as a population of resident macrophages called alveolar macrophages. So really, as I mentioned, a number of groups had showed some pretty important work about conditioning of these soils, and specifically about conditioning of the lung, including Jeffrey Pollard's group, describing a role for these monocytes, these CCR2 monocytes, in promoting early lung colonization and metastatic outgrowth in certain tumor types. So there was really tumors like breast, colon cancer, things like that. And nobody had really looked at it in osteosarcoma. And so really a lot of this work comes from my PhD work, and then subsequent ongoing collaborations with Dr. Steve Dow's laboratory, where we started to look at this process in osteosarcoma. And just to kind of summarize our findings, we observed that both canine and human osteosarcoma cells significantly overexpressed this monocyte chemokine, CCL2. which we confirm via ELISA measurement of CCL2 secretion here shown in these lower graphs in these cell lines. And when we looked in canine osteosarcoma lung metastases and we performed immunohistochemistry, we could see significant enrichment of these monocyte infiltrates in these tumors. So these are these cells here shown in red, infiltrating this lung metastasis here. And that tumor cells within these metastases were producing abundant amounts of that chemokine CCL2. So that's this red staining within the cytoplasm of these tumor cells. And again, going back in vitro, working in some human osteosarcoma cell lines, we showed that conditioned media, right? So soluble factors of these osteosarcoma cells could result in robust recruitment, in vitro recruitment of monocytes. So this is just using traditional transwell monocyte migration assays and primary human donor peripheral blood monocytes. And so at this point, we really wanted to find a compound, if you will, to target this process, right? To target and block this monocyte recruitment. And so Steve's group had performed some in silico modeling of that CCR2 receptor that these monocytes use to move around the body. And we screened a library of approximately 400 already FDA approved, off patent, cheap and known to be relatively safe small molecule compounds to try and find drugs that would bind and inhibit this CCR2 receptor. And so we came up with a short list of compounds, and one of the molecules on that list was losartan, or also known as Cozar. So losartan is already being given to millions of Americans around the country. It's used to treat high blood pressure or hypertension. But there was also some work already in the literature, primarily in diseases like atherosclerosis, which had showed that losartan might have these immunomodulatory effects, and specifically blocking monocyte accumulation within those atherosclerotic plaques. And so we move forward with losartan as our kind of potential candidate for a CCR2 inhibitor compound to block this monocyte recruitment process to these osteosarcoma metastases. So again,
sorry, can I just ask you a question? So when you were looking at is it the CC, sorry, can you go back like 2 slides? Was it the CC? Yeah, the CCL2, CCR2. Were you looking at cells when you're saying that there are cells that are promoting the lung mets? Are these cells that survived after chemo that you're thinking then go on to metastasize? Or sorry, can you just
great
cells those are?
Great question. Yeah, so these are and again, this is some work by other groups, but I'll show you some data in just a minute. But yeah, very, very early on in the metastatic process. So when you're getting, microscopic or micrometastases forming in the lung, so tumor cells just seeding the lung, you have this enrichment of CCL2 production that is recruiting these monocytes out of the bone marrow into the blood, and they're infiltrating the lung. And so they're helping these cells colonize the lung and survive within the lung, and then, turn into overt metastases later on. So these tumor cells are likely surviving adjuvant chemotherapy. They're already seated in the lung, and then they're leveraging these cells as somewhat of a support system, if you will, to help them survive that stressful process of colonization until they can kind of grow into essentially an organ, right? A large gross metastasis at that point.
Got it. So these are the cells that not have only survived the chemo, but have also started to seed in the lung.
Yeah, they've started to seed, and they're leveraging these monocytes to help them survive and grow in the lung.
Thank you.
Yep. So indeed, we started to screen losartan, and that's just denoted with the L here in these graphs, or LOS. But started to screen losartan again to see if it could inhibit this tumor-induced monocyte recruitment, and specifically the CCL2-induced monocyte recruitment by these osteosarcoma cells. And so what you can see here, again, these are in vitro monocyte migration assays. But again, with these 4 different human osteosarcoma cell lines, we would see potent monocyte migration. And when you pre-treated these cells just for 1 hour with clinically achievable concentrations of losartan, we could see at least a 50% reduction or greater in this monocyte migration, whether you were using primary human monocytes derived from blood donors or working with a monocyte cell line, which we use is called THP1 cells. And here on the right are just some representative images of that monocyte migration. Each of these small blue dots is a monocyte, and you can see these blue dots are decreased here with increasing doses of losartan in vitro. So at this point, we move losartan into some in vivo monocyte migration model. So we would use a vaccine adjuvant where we could inject this into mice and cause that inflammation and CCL2 production to recruit monocytes into the draining lymph node. And then more importantly, obviously to us, was to look at metastasis-induced monocyte recruitment. And so in these assays, what we would do is we would inject tumor cells, in this case, green or GFP labeled tumor cells, IV into the veins of mice, and they would seed the lungs in these very small, early microscopic clusters. And then we would wait 72 hours, typically, or 3 days, and then harvest these lungs and look at tumor cell colonization and then monocyte recruitment into these lungs. And so what you can see in control healthy mouse lungs, there's a very low percentage, roughly 1% of these monocytes within the lung. When you seed tumor cells, again, microscopic tumor cells in the lung, we have a nearly 6-fold increase, up to 6% of these monocytes here in the lung. But when we treated these mice once daily with losartan, or when we ran these studies in mice that were genetically knocked out for that CCR2 receptor, we saw a very significant reduction in this monocyte population in the lung. And again, that's captured here in these images in this central panel. So again, tumor cells in green, the monocytes were able to stain, immunohistochemically for red. And you can see these cells co-localizing together, right? This really intimate association of these monocytes with this early metastasis. And again, treatment with losartan could significantly block this. And again, one thing we observed early on, so again, we were really just more focusing on the mechanism at this point of losartan blocking this monocyte recruitment. But when you image these whole lungs and looked at these green tumor cell micrometastases, you could see that they were significantly reduced in number, even at this early 72-hour time point. So removing this monocyte population really seemed to slow or reduce this micrometastatic burden, if you will. And again, you can see these clusters of green cells here in the control, and there's far fewer in both losartan and the CCR2 knockout mice, and that's just quantified here in this lower panel. And so at this point, we were pretty confident that we had a good candidate CCR2 inhibitor or monocyte blocking drug. And so we employed it in some longer term metastasis growth assays here. And so here we're dosing these mice once daily with losartan, and we're monitoring lung tumor growth using IVUS bioluminescence imaging over time. And so again, you can see over the course of 3 weeks, we're really getting slowed metastasis growth, right? So we're not completely inhibiting metastasis formation, and I think those images on the previous slide showed that, but this growth is significantly reduced as compared to control mice. And again, there's some images at euthanasia of these mice here, where you can see all of these blue nodules in the lungs are the metastases, and then on the losartan treated animals, yes, there's still metastasis as you see in the IVUS images, but they're far fewer in number there. And importantly, we saw sustained, this reduction in metastasis was associated with sustained monocyte blockade. So again, not just looking at 72 hours, but looking after 3 weeks of dosing and after, formation of these larger multiple gross metastases in the lungs, even still, this losartan was able to significantly reduce the number of monocytes in the metastases of these mice. And so again, that's quantified here on the top with flow cytometry, and then on the bottom with actual labeling of these monocytes in the metastases. So on the left is control, and on the right is losartan treatment, and either red or green is denoting this monocyte population. And again, with treatment, it's significantly reduced. So at this point, we were quite confident that we had a pretty solid therapy to move forward, but we also knew losartan wasn't a silver bullet, right? I just showed you that these mice were still developing metastases and they were still growing, and so we wanted to look for another therapy that we could combine it with. And at the time, the only tyrosine kinase inhibitor approved in veterinary medicine was this multi-kinase inhibitor, toceranib. So toceranib is functionally equivalent to the human drug sunitinib, which is approved for use in human osteosarcoma patients. And specifically, we felt this was kind of a rational complementary strategy because there's a lot of literature using sunitinib, not just for its kinase inhibitor, direct anti-tumor effects, but also as a tumor microenvironment modulator. So people had showed that through inhibition of STAT3 signaling, that sunitinib could target other myeloid cells like myeloid-derived suppressor cells and other cell types of that microenvironment that were known to promote tumor progression and metastasis and immune suppression. And so that was the combination we moved forward with, and was also, again, based on some of our own mouse data. So we showed when you dose mice with lower doses of these compounds, but gave them in combination, you could see a significant reduction in osteosarcoma lung metastasis burden in the combination treated animals versus just single agent treated animals here. And so, naturally at this point, again, we felt we had enough preclinical evidence to move this forward, and being veterinarians and working at the largest animal cancer center in the country, we wanted to assess this in a clinical trial in dogs and in dogs with spontaneous lung metastatic osteosarcoma. And so just briefly, for those of you that perhaps aren't familiar with the comparative oncology approach, myself and many others in the comparative oncology community strongly feel that dogs with spontaneous osteosarcoma are perhaps the single best model for a human solid tumor counterpart. And this is really due to a number of factors and a lot of work by people that have come before me, Sean Khanna, Steve Withrow, a number of individuals. But dogs and humans have dogs and human osteosarcoma have significant genetic and biological similarities, and these include a number of shared driver mutations, natural evolution of drug resistance and lung metastasis development. And then I think, almost perhaps most importantly, dogs will develop osteosarcoma at least a 20 times, upwards of, certain papers cite upwards of 80 times higher frequency than people, right? And it's also with the shorter, dogs have a shorter lifespan. They also have a shorter clinical course of disease, right? So you're going to get more rapid vetting of these preclinical discoveries or preclinical therapeutic candidates in the canine model, but also, in my opinion, with a higher predictive value, right? So if something, we know things can work in mice, but if something's working in the dog, we really feel strongly that has a better shot of working in people. And so that's really why we take this approach. And so this is out of Sean Khanna's lab, previously at the NCI. And this is just gene expression data from both human and canine osteosarcoma primary tumors, and just the 30,000 foot view here of this heat map, which you can see. So blue, the little blue dots at the top of the graph are the canine tumors, and the red dots are the human tumors. And so when you allow these to cluster, right, if they were different, they would cluster separately. But what you see is all of these intermingling, meaning that, in terms of gene expression, in terms of the whole genome or transcriptome of these tumors, the general patterns of gene expression are very, very similar across canine and human tumors. So really getting back to that genetic similarity I've mentioned.
I'm just curious so now we know a little bit about like some of what the more common mutations are.
Yes, yeah.
Human osteosarcoma, are those similar in canine? So, MYC, ATRX, RB1.
Absolutely. Yeah. So, there's a number of publications out on that. So Heather Gardner and Cheryl London have a publication. Some people right here from the Flint Animal Cancer Center that I'm on as a collaborator as well, Don Duvall's group, have shown, that the highest frequency mutation is P53, but RB1 mutations were also quite common, and a pretty high frequency of MYC gene amplification as well. So again, a large number of those drivers seem shared, but that's still an active area of research within the comparative oncology community for sure. So we moved forward with this trial in dogs, again, with spontaneous lung metastatic osteosarcoma, and this was losartan combined with toceranib, which again is equivalent to the human drug sunitinib. And we initially started this trial at a losartan dose of 1 mg per kg because that was a recommended dose for treating dogs with high blood pressure. And we enrolled initially 8 dogs at this low dose, and while we saw some stable disease, we really didn't see a strong clinical benefit. But we measured losartan drug levels in the serum of these patients or in the blood of these dogs, and realized we were well below those levels that we had observed in mice and observed in some of our in vitro assays. And so at that point, we escalated the losartan dose approximately 10-fold, much to the dismay of our hospital pharmacists and in clinical trialists. They were afraid these patients would have a significant drop in blood pressure and perhaps faint. But They did not. In fact, it didn't affect their blood pressure at all. It's quite well tolerated. And most importantly, when we did, we really started to see some significant clinical benefit of this high dose losartan combination. So now we observed a 25% objective response rate. So, 25% of these dogs actually had a partial response. Still a large number of dogs, or 25%, with stable disease. And so an overall clinical benefit, if you will, and this is durable, stable disease out greater than 8 weeks in these patients. So this was an overall clinical benefit rate of 50%. And here's just one of our patients here, where you can see this lung metastasis circled in red, and then 8 weeks post-therapy, this particular nodule had completely regressed here. So I think I got, just another example here. This is our 5th patient in this high dose cohort. This is a little more poorly defined nodule, but again, I think you can still appreciate this here. And this patient had, again, a really strong partial response where this mass was still present, but significantly reduced in size and in density. And this is just some images from the necropsy or autopsy, if you will, of this patient. And again, this was the only lung metastasis we could find there. So this point regressed to microscopic disease. And so again, this high dose combination was observed with a significant prolongation of both progression-free and overall survival here when you compared it to both the low dose losartan combination and then that kinase inhibitor alone, toceranib. So, at this point, we were pretty excited about this data. We felt like this was a pretty good therapeutic signal. And we were presenting this data locally at our own Children's Hospital, Colorado, and some of our collaborators out at the University of Colorado. And again, these clinicians are treating human patients with osteosarcoma, and they got quite excited seeing this benefit here for metastatic disease. And so ultimately, this canine data did lead to direct translation of this combination to human metastatic osteosarcoma patients. So we currently are enrolling in a phase 1 trial, and this is being conducted both at Children's Hospital Colorado, the adult hospital at University of Colorado, and then Children's Healthcare Atlanta. And this is driven by Kelly Falk at Children's Hospital Colorado and Thomas Cash at Children's Hospital Atlanta. And so thus far, we've just enrolled. Again, we're looking to push that dose of losartan, similarly to how we did in the dogs, right? To where we went from that 1 mg per kg to that 10 mg per kg dose. We want to take this dose as high as we can in these patients, while obviously still hoping that it's well tolerated, and we expect it will be based on the canine data. But thus far, we've enrolled the first 3 patients at this lower dose level, or dose level 1, and we've enrolled 1 patient at dose level 2. And so what we are able to do in these patients, our lab's able to do, is monitor that population of CCR2 monocytes in their blood through flow cytometry, or in this case, cytoph, or cytometry by time of flight. And so you can see here, there's a large percentage of these cells are denoted in red. They have high expression of that CCR2 molecule, shown down here in green in that cluster. And when we look at after 14 days of losartan treatment in these patients compared to their pre-treatment blood levels of these monocytes, in all 3 of that first dose level 1 patients, we have been able to significantly decrease these cells. And so, we're glad to see that we're really hitting that pharmacodynamic target of CCR2 monocytes, if you will, and we're optimistic. We haven't seen any responses yet, but we are optimistic as we kind of escalate that dose, that hopefully we'll see some of these patients at least see some clinical benefit from this drug combination.
That's super exciting. I'm curious, for the canine trial that preceded this, were you able to do any correlative studies to just see if there are any biomarkers for response for the patients that did respond?
Absolutely, yeah. So one of our markers, the best marker of response was another chemokine called IL-8 or CXCL8. And this molecule actually has a lot of overlap in a way with some of the functions of CCL2. So CCL2 is recruiting monocytes. IL-8 or CXCL8 recruits neutrophils and could recruit, granulocytic, what are called granulocytic myeloid-derived suppressor cells. And so those dogs that had higher baseline levels of IL-8 responded better to the therapy. And then those patients that had a lower fold increase in IL-8 post-treatment, so 1 month post-treatment, also lived much longer than those patients that had increasing levels of IL-8 on the trial. and that was you know, irrespective of disease burden in the lung, we were able to account for that. So that's actually informing another trial that we are running in dogs, and Steve Dow and Cheryl are running this and driving this, is looking at that combination with an IL-8 blocker as well. So okay, so like I said, we're really taking this kind of three-pronged approach, and so I want to shift gears now. And in the past few years, our lab has shifted our focus onto the role of lung fibroblasts within this osteosarcoma lung metastatic niche, and this has really been driven by a graduate student in my lab, Eric Palmer. And so this shift in part, there it is. So this shift in part really arose through an observation we had of significant enrichment of fibroblast-derived matrix proteins. So these are extracellular matrix structural proteins present within human and canine osteosarcomas, and then an associated increase in integrin signaling. So integrin receptors are cell surface receptors that will integrate signals from matrix proteins into the cell. And so we observed a significant enrichment of this here, which is shown in these two gene set enrichment analysis plots here for canine osteosarcoma cells on top and human osteosarcoma cells on bottom and B. And this really led to a lot of the supporting data and some of the proposed work for the OutSmarting grant that I'm about to talk about now. And so, you know, we had performed RNA-seq on both human and canine tumors and observed this increase in integrin focal adhesion kinase signaling. And so I'm sure many of you are aware, but focal adhesion kinase is a non-receptor tyrosine kinase that really integrates that integrin signaling and can drive a number of direct tumor promoting effects in other cancer types. That had never been described really in osteosarcoma, but in other tumor types, pancreatic cancer, ovarian cancer, it can drive cell survival, proliferation, and migration. And so, again, had not been looked at in osteosarcoma. And so when we observed this, we really wanted to kind of follow this lead. And so we were able to confirm some of our gene expression data with Western blots of human osteosarcoma cells shown here, where even in the absence of serum, right, they have constitutive activation of this FAK signaling. So this is phosphorylated FAK, or the active form of FAK, here in the top row. And you can see these dark bands here corresponding to these negative columns. So all four of these cell lines had a pretty high level of FAK signaling. And then again, when we go back into our canine patient osteosarcoma lung metastases and perform immunohistochemistry, you can see robust FAK labeling within the cytoplasm of these cells. So all of this brown staining here. And so to target this FAK signaling, we began working in vitro with defactinib. So this is a focal adhesion kinase inhibitor. It's currently in phase 2 clinical trials, again, for a number of other solid tumor types, mesothelioma, melanoma, pancreatic cancer, but has not been looked at in osteosarcoma. And so you can see, again, in vitro treatment with this FAK inhibitor, both canine and human cell lines significantly inhibited cell survival and proliferation. And this was at relatively low concentration, so nanomolar concentrations of these drugs. So they were quite sensitive. And again, we were getting strong target modulation, right? So now we were able to reduce levels of this phosphorylated or active form of FAK. And then when we put this drug into a mouse human xenograft osteosarcoma metastasis model, again, we could see that just single agent FAK inhibitor therapy significantly extended survival of these mice. So a good anti-metastatic activity just as a single agent. But was even kind of more interesting to us was that some of the extrinsic, again, tumor microenvironment effects of this FAK signaling and how it may be playing a role in shaping that osteosarcoma microenvironment. So again, we had that RNA-seq data, we observed enrichment of this FAK signaling, and when we split or dichotomize these tumors, both canine and human, into those tumors with enriched FAK signaling versus those tumors that had low FAK signaling, the FAK high tumors had this enrichment of these immune suppressive gene signatures. So enrichment of immune suppressive cytokine production, monocyte and macrophage abundance, and T and B cell suppression within these FAK high tumors. And so again, this was something that was of extreme interest to us, kind of linking this signaling both with these direct pro-tumor effects within the osteosarcoma cells themselves, but also perhaps a role for FAK in shaping that tumor microenvironment. And again, we were able to support this data with, again, our canine osteosarcoma samples. So going into canine osteosarcoma lung metastases, we were able to perform single cell RNA-seq analysis. And what we observed with that analysis is that FAK expression, which is denoted here in purple in this top left panel, was primarily restricted to tumor cells, and again, those matrix producing fibroblasts, or those lung fibroblasts. So that's quantified here on the bottom. The two highest expressors of FAK within the lung osteosarcoma lung metastasis was the tumor cells themselves, and then these fibroblasts. And then importantly, again, our friend CCL2 here. And so these FAK expressing fibroblasts were the highest producers of that CCL2 chemokine here. So suggesting a possible connection there. And importantly, we were able to confirm this in human primary donor-derived lung fibroblasts as well. So again, leveraging those osteosarcoma-derived soluble factors, those exosomes again, we were shown that these human fibroblasts were able to take up these human exosomes here denoted in red. And when they did, it really primed these fibroblasts to promote or to produce a number of pro-metastatic cytokines and chemokines. And again, CCL2 being one of the most significantly upregulated chemokines in response to osteosarcoma cell priming of these fibroblasts. And so again, with this really to us was connecting the dots here in terms of how these fibroblasts perhaps may serve as a linchpin to start that monocyte recruitment process into the lung in osteosarcoma. And so again, moreover, kind of using these same assays of where we could prime these human lung fibroblasts with these osteosarcoma soluble factors, or these exosomes, we observed that these exosomes from all 4 of our human cell lines would promote proliferation of these fibroblasts, quantified here. And when we treated, and it would also again promote production of that chemokine CCL2. And when we treated with this FAK inhibitor, we were able to suppress these responses, right? So we were able to suppress this fibroblast proliferation with a 1 micromolar dose of defactinib, and you can see that reduction in fibroblast number here in these images. And again, we were really able to significantly shut off that CCL2 production by these fibroblasts with defactinib. And so this was important to us. So we felt like now that this FAK inhibitor, not only could it directly kill these osteosarcoma tumor cells, but it also was a tool for us to target these lung fibroblasts, which we felt like were promoting this monocyte recruitment. And so targeting these lung fibroblasts, we think is a really critical way to start to get at, to target these osteosarcoma lung metastases. And this is some of the data of why we think that. So when we take our human osteosarcoma cells and we use this in vitro assay of co-culturing them, again, with these are primary human donor-derived lung fibroblasts, and you stress the cells as if you're trying to mimic that early metastatic colonization of the lung process, right? When few cells are arriving in that lung. So we stress them by serum starving them in this culture assay. You can see that the osteosarcoma cells alone, just in the plastic dish, do not survive. So that's the top line here in green, and this far bottom left column here. So they can't survive and handle that serum starvation stress. But when you co-culture them with these lung fibroblasts, they can, and they start to proliferate. And when you prime these lung fibroblasts with these exosomes and get that CCL2 production and IL-8 and IL-6 production increased, you get an even better survival benefit. And that's just captured here in these images in the bottom right. So you can see these osteosarcoma cells are in red, and they're surviving in this co-culture with these lung fibroblasts, and they're growing even more robustly when you have this priming of these fibroblasts and get those chemokines and cytokines going. And so that really brought us to this first aim of our OutSmarting Osteosarcoma work. And it's the foundation, which is determine whether FAK inhibition through, again, combined targeting of osteosarcoma cell survival and proliferation, targeting of these fibroblasts, and then macrophages add significant clinical benefit to losartan-toceranib immunotherapy. And again, running this trial in dogs with spontaneous lung metastatic osteosarcoma. And so thus far, we've optimized defactinib dosing in dogs. So that was a bit of an unknown, and obviously oftentimes is when you're moving from mouse to dog. And so doing this in healthy dogs, we've seen that it's well tolerated, and that in the blood of these dogs, we can get defactinib drug levels that significantly exceed those required for that in vitro target inhibition and immune modulation that I just showed you. And so the trial is open now, and we're just starting to enroll patients, and we hope to accrue roughly 1 patient per month into this study. And these, again, will be dogs similar to human patients, right, or how you would run this trial in human patients. So they will have had prior resection of their primary tumor. They'll have received adjuvant chemotherapy, and then they'll have eventually relapsed or failed that therapy with lung metastatic disease. And that's when they will be enrolled into our trial, receiving this triple drug oral immunotherapy. So defactinib, losartan-toceranib, all orally delivered, can be given at home. We can send these patients out the door, which I think is another big plus there as well. Okay, so in these last 5 to 8 minutes or so, I just want to touch on some of the work we're doing with this third cell type of interest. So this final cell type we're interested in, the lung metastatic microenvironment, is the resident alveolar macrophage. And so our interest in this cell is really twofold. So one is, could these cells serve, these are resident cells, they're residing and living in the lung already, ever before the arrival of tumor cells or even the development of cancer in a patient. So they don't have to be recruited there like these monocytes. So could these cells perhaps serve as another source of these monocytes or tumor-associated macrophages, which is what these monocytes differentiate to? But then more so, we're really interested in their kind of normal physiologic role as sentinels of the lung microenvironment and really maintaining homeostasis or immune homeostasis within the lung. And so based on that sentinel role, we kind of hypothesized that these cells may serve as a key target or cellular target of those osteosarcoma-derived soluble factors, those exosomes, other things that are being released from that primary tumor and priming that lung soil to help facilitate this metastasis process. And so, again, this is some work. This is being driven by Laurel Haynes, a DVM PhD student in the lab, going back to our mouse models, but using those human osteosarcoma exosomes that we isolate. When we inject those exosomes into these mice, we see this selective homing to the lung and accumulation of these exosomes in the lung. So we can, again, fluorescently label these cells and monitor their tracking with in vivo imaging in these mice, and again, mirroring that clinical picture of that metastatic tropism for the lungs. These exosomes are also preferentially accumulating here, and that's quantified here in these 4 graphs. So this is the in vivo imaging in these 2 middle, where 143B is our osteosarcoma exosomes. And there's an increase in the lungs, and it's preferential to the lungs versus these other solid organs here. And then again, this is by flow cytometry over on the right, and we see this increase in exosomes within these cells. And more importantly, the cellular target within the lung appears to be, at least in part, the alveolar macrophage. So, sorry, there we go. So again, when we digest these lungs and figure out who is taking up these exosomes, we can do that by flow cytometry, and we see this population of 11B positive or 11B low, CD11C high cells in the lung, and that's characteristic for an alveolar macrophage. And even more importantly, we can measure those chemokines in those lungs, right? Some of those same chemokines I showed you are changing in response to the osteosarcoma exosome priming of the human lung fibroblasts. We see that in the mouse, and we see it, selectively or better able to capture it when we look in the airways of these mice. So these alveolar macrophages reside in the airway spaces of the lungs often, and so we can collect fluid from those airways, which is called bronchoalveolar lavage fluid. And when we do that and compare it to just collecting serum or blood, we see this preferential increase here. So again, suggesting that kind of potential sentinel role for the changing lung microenvironment with osteosarcoma metastatic progression for these cells. And so that really brings us to our AIM-2 work and another overarching hypothesis of the lab, which is proposing kind of applying a rather long-standing diagnostic technique for lung disease, which is bronchoalveolar lavage, as a new immune monitoring approach in our canine metastatic osteosarcoma model. So we're not necessarily proposing that this would be moved into human patients and done as part of their workup or monitoring response to therapy, but more so as a tool that we can uniquely leverage in the dog model again, right? Another valuable aspect of that model to learn more biology about this lung environment or this soil and how it's changing, and specifically how it might change in response to therapeutic intervention. So again, we can enroll these patients, we can flush this fluid into their airways, and when we do, we will retrieve these alveolar macrophages, and we can measure changes, immunological changes in those cell types. And so really to kind of just to begin to understand this process and to demonstrate our feasibility to do that, we've been enrolling healthy dogs as well as dogs across the phases of osteosarcoma progression. So healthy dogs with primary localized osteosarcoma, and then dogs with lung metastatic osteosarcoma, and been performing single cell RNA sequencing on bronchoalveolar lavage fluid cells. And so thus far, we've only done 4 to 5, I think 4 healthy dogs and 5 primary osteosarcoma, so localized osteosarcoma bearing dogs. But even there, we've observed some very interesting changes. So we've been able to identify 40 different cell types within this bronchoalveolar lavage fluid. So suggesting this fluid perhaps could be a very good surrogate for that lung metastatic microenvironment. And then of those 40 different cell types, 16 of them are macrophages, and 11 of these 16 macrophage populations are significantly enriched in osteosarcoma bearing dogs. And that's shown here in blue in this panel B here. So osteosarcoma dogs are purple, healthy dogs are green. Each one of these dots is a cell. And so if you compare B to A, the macrophage, those 16 macrophage clusters are primarily located up here in this larger top cluster, and many of those dots are purple, right? So coming from these osteosarcoma bearing dogs. And again, kind of paralleling our mouse model data here, we've observed that CCL2 levels within that fluid of the airways is better correlated with that macrophage density than CCL2 levels without in the periphery, right? And then even also more importantly, some of these clusters of these monocytes and macrophages seem to be correlated with FAK expression. So remember a few slides back, I showed you that robust FAK expression in the single cell RNA-seq of an actual metastasis. We also observe FAK expression when we sequence these bronchoalveolar lavage fluid cells. And when you look at FAK expression and whether it's correlated with these myeloid cells, we see that increasing FAK is indeed correlated with some of these clusters. So again, suggesting that FAK signaling could sort of be the linchpin that starts to drive that cascade of CCL2 production from those fibroblasts that's bringing in these macrophages here. But again, this is early data, but our hope is to, and this is built into our trial, is we're going to do this bronchoalveolar lavage sampling and sequencing pre-treatment in these dogs that are receiving this triple drug therapy, and then do it again at 8 weeks post-therapy, and hope to kind of learn something additional about indicators of response or correlates to response that we wouldn't necessarily pick up in the peripheral blood. And so really that kind of brings me to the summary here is we're really trying to understand how hyperactivated FAK signaling can kind of drive this fibroblast and macrophage rich metastasis promoting microenvironment in the lung for osteosarcoma, and then how we can leverage defactinib and then our pre-existing immunotherapy backbone of losartan and toceranib as this triple drug immunotherapy to target this whole process. And again, to determine if sampling of that alveolar space can serve as a better surrogate of that lung microenvironment and changes in response to therapy than traditional immune monitoring, which is done in the peripheral blood. And so I think we could, perhaps stand to learn some additional biology about lung metastasis of osteosarcoma and perhaps response to therapy with that approach. So with that, that's all I have, and just some acknowledgments here and the group, and really this is a big joint effort. I hope I captured that. We have a really great clinical trials team here at CSU that runs these canine trials. Obviously, our human collaborators running these trials and our human patients in Colorado and Atlanta. Steve Dow's group, as I mentioned earlier, with a lot of the monocyte work as well. So.
Thank you. That was a really great overview. Everyone, please feel free to add any questions to the Q&A, and we'll make sure to ask Dr. Regan. And I think Vicki might have had a question to kick us off.
Yeah,
Okay.
Just the first question. What are the side effects for human patients for, I feel like I pronounced it wrong. Losartan, yeah, sorry.
Yeah.
And
Sunitinib.
Yeah. Yep.
Yeah. So far it's been really well tolerated. So we haven't seen any dose limiting toxicities in those first 3 patients, and really, low grade, I think grade 1 or 2 adverse events. So with losartan, there's not much that we're aware of that would be a bad side effect. I think with sunitinib, there are a number of other side effects. One perhaps even being you can see cardio toxicity, and so patients that have received, doxorubicin prior to sunitinib therapy, we definitely have to watch for that. But yeah, the losartan is well tolerated. It doesn't seem to reduce blood pressure or cause any sort of lethargy or anything associated with that, at least thus far.
And what about the defactinib, Dr. Regan? Is that
Yeah.
well, is it complete? Well, I guess both a tolerated thing, and is it purely investigational drug at this point? Has it been indications for it, or?
Yeah. As far as I'm aware, it's still an investigational new drug. I think it's definitely in phase 2 trials. It's being, I think, used often too with like MEK inhibitors that target the MAP kinase signaling pathway, but it seems to be, based on a published study of a phase 1 trial, well tolerated. So again, all, I think, grade 1 or 2 dose limiting tox, grade 1 or 2 adverse events, one of which is like an increase in a liver enzyme, which was non-clinical. So that data, that safety data was very, very promising. I don't think they've published any of the phase 2 trials that are ongoing right now, but they're definitely ongoing in pancreatic cancer, mesothelioma, and I believe ovarian cancer. And similar to our approach, they're using these FAK inhibitors in combination with checkpoint inhibitors, so PD-1 inhibitors and things like that. They really think that targeting FAK through targeting fibroblasts, through targeting macrophages, could potentiate immunotherapy response, and we feel the same. We think that targeting FAK could potentiate our losartan-toceranib immunotherapy.
Great. And I had another question that, of course, just like escaped my brain, but I was just curious for in general, because we've seen, actually, sorry, I know what my question was for the lavage. Is that something that is kind of new in the study, using that, or has that been used before as kind of the surrogate to investigate the tumor microenvironment, or is that something that's kind of new with the study?
Yeah, it's completely new. I think the closest thing people have done airway sampling and perhaps even lavage in lung cancer patients, where you can't get you got it's an inconclusive biopsy of trying to get tissue. So these are patients with primary lung cancer, and so they're looking for tumor cells, or they're doing PCR looking for specific gene mutations that are known, associated with lung cancer, to try and confirm a biopsy or confirm some of the imaging that is suspect. But nobody's done it to monitor cancer like this and therapeutic intervention, especially in dogs. And again, I really think it could be a unique tool in the dog to try and learn more in this model, and as we're testing these new therapies, perhaps.
Yeah, no, I think that's really, like, for all the reasons that you set up in the first initially about kind of all the benefits of the canine trials, like this is just like another one, right, where it's like you can better interrogate that tumor microenvironment. Yeah,
exactly. And we're hoping to really try and benchmark it against the blood. Yes, we're always going to be collecting the blood as well, and seeing, what's similar, what's different, things like that. So.
We just had another question come into the chat. Is this clinical trial medication limited to a certain age of patients?
Yeah, great question. I think it is. I think we limited the human phase 1 trial up to patients that are 26 years old. And I think, obviously the human medical oncologist would be able to comment better on that decision of why we went with that age group of patients there. But I think, we'll enroll up to age 26.
Okay, this was really great. We probably just have a couple minutes left to wrap up, but really appreciate and thank you for just like walking through that so clearly. And it's great to kind of see how one trial has just kind of continued, you've been able to just build, like
Yeah,
from going into a human trial, but then also building and kind of looking at another combination. So really interesting kind of to see how that's progressed.
yeah, thanks again. Thanks for having me. Obviously, thank you for funding the work. And like you said, yeah, we're really kind of hoping to just keep going back to the, maybe not the drawing board, but of something a little farther along there to build on this and run more therapies through our canine patients and hopefully get some things that stick and make a difference. So that's obviously the ultimate goal there.
Yeah, and we'll
Yeah.
look forward to checking back with you in a few months once
Yeah.
you have some results from this study, which I'm sure will be, I'm optimistic for some really good results coming out of the
We are
study.
we are as well. So
All right, well, thank you so much for joining us today on OsteoBites and for making it better for both canine and human osteosarcoma patients. And more information on this and all OsteoBites can be found on our YouTube channel, also on our website, mibagents.org. And you can always listen at your favorite podcast place. And next Thursday, please join us. We're going to be talking with Dr. Karun Sharma, who's a director of interventional radiology, and Dr. Eran Kim, who's a member of the solid tumor faculty and an associate professor of pediatrics. And they're both from Children's National Hospital, and they're going to be discussing interventional radiology and HIFU perspectives for osteosarcoma. And the registration link is in the chat if you're interested in joining that. And you can find the lineup for the next few months for OsteoBites on our website. And if you ever have any ideas for future topics that you'd like to hear about on OsteoBites, just please let us know at events@mibagents.org. And thank you again to Dr. Regan for spending an hour with us today, and also to Vicki. And we're looking forward to having you back on OsteoBites in maybe a year or so to hear about those results. And thank everyone for joining us on OsteoBites today. And we hope to see you next week when we talk to Dr. Sharma and Dr. Kim. Thanks so much, everyone.
all right. Thanks for having me.